Hello, everyone. We are now starting Laboratory 2 in the class. The basic tenets of Lab 2 are to try to understand the processes of meiosis. Meiosis is really the first part of sexual reproduction, and that entails the production of gametes that go into fertilization, both in males and in females. What's important for us as evolutionary biologists is try to understand that how is variation established in this process of meiosis. And when we think about meiosis, there's an idea that we should have in our head is that that's where all evolution occurs and in no other place. And in fact, the thing about it, if we're historical detectives trying to think about what Darwin was thinking about, in his time when he was developing the theory of natural selection, he understood and he saw this that every single species in the world around him produces variation. There are no exceptions. And Darwin understood this as a tenet to natural selection and evolution. He didn't know how it occurred. He didn't understand meiosis simply because during his period of time, the technology wasn't developed to be able to explore the cellular terrain and high definition and high magnification microscopy. So we'll be learning about that in this lab. Now, previous in the lab, we looked at, or in lab 1, we started to get some of the basic tenets of how DNA operates, the idea of transcription and translation, and also a couple of ideas of how mutations occur, right? And those go on to produce proteins which are different than the ones that are existing in one generation, and you're propagating changes which will lead to other sorts of changes, right? But it's how those mutations and where those mutations get laid down at that is the centerpiece to this lab in meiosis. Okay. So the first things to think about are what's important in all the technical stuff that you're looking at. In some ways, it's impossible not to do sort of a basic biological sort of course routine in this to sort of understand it, because you have to understand some technical aspects. But many of you are thinking about what do I need to know for an exam, and that's a really good question. What should we be focusing on? The big ideas. Now, when we talk about the idea of meiosis, we're talking about the idea of taking a specialized cell. Okay, so we're going to be talking about a specialized cell, a sex cell, right? Not the regular somatic cells of your body, not like a fingernail cell or an eyeball cell, but those that are within your sexual reproductive system, and males in the gonads, right? Females, of course, somewhere in the ovaries. And as you're producing gametes, these primary sex cells have to reproduce themselves. So we think about in cellular reproduction, what must occur is DNA replication. Now, we've seen that in the previous lab. What we have to be able to do is anchor this concept of DNA replication, right, into the process of meiosis, because it's in the replication. That one moment is where all evolution happens, and no place else. When you're producing your gametes, it's mistakes in that replication process, right, that are passed into future generations. Now, the process of meiosis is the process of DNA replication, and subsequently trying to take the mistakes that are made in DNA, right, the mutations which we've seen, and randomize those, statistically randomize those as much as possible into the gametes, the smaller sex cells that go into reproduction, right? Randomize everything. And that's really what meiosis is about, okay? We begin with reproduction with mistakes, but replication of DNA with mistakes, and then randomizing those mistakes as much as possible, mixing them around like a salad into all sorts of different gametes. So it will increase the amount of variation as much as possible. So if we can keep it simple, and keep those basic concepts, I think it makes the technical stuff sort of easy, because you kind of see the longer, sort of the outcome a little easier. Okay. Now, as far as terminologies, I really want you to focus on a couple of ideas. One is the idea of independent assortment. This is Mendel's principle of independent assortment. And this is really where most of all evolution happens at. So independent assortment is going to be dealt with in this lab a lot, not only in the workbook, but also in the films I've got below for this laboratory, right? Laboratory 2. Now, one of the films I have below is from, I guess, America's favorite video high school teacher, Mr. Anderson. And surprisingly, as much as this is built for a high school sort of class, it's really quite good. And I think it's really quite informative to sort of explain what meiosis actually is. right? And we're going to return to another video by Mr. Anderson. And what he does is he does something with some beads, a very interesting little prop, to sort of show you not only how the process of meiosis works, but how this idea of independent assortment. And really what that is once we get division, replication of the DNA and the mistakes are made, it's independent assortment that shuffles them into the different gametes. So it's going to give you a really good sort of a bead description. I've tried to do this myself. I have to tell you, the ones that I've done, they're pretty much exactly the same as Anderson's video, but I think he had the edge of me a little bit on what he did, so I'm going to put it up there. I'm going to let him outbetter me on this one. All right. Now, the last video segment we have down here deals with the idea, and we've sort of started to build on this idea, of the idea of some genes being more dominant to others, the idea of dominance and recessivity. Right? So we'll be in this lab. We'll be looking at who discovered this, Gregor Mendel, and how this idea of dominance and recessivity is not what it may seem. For example, when people think about, this idea of recessive genes, they always think about negative consequences of these things, right? Like some diseased condition, because, in the public consciousness, we have to face it, we're human beings. We're always looking for the negative aspects. When you say bacteria, everybody, bacteria, harmful disease, what we don't know is we don't realize that we have 9 trillion bacterial cells living within us right now, and they're helpful. And without them, you all would be dead within a week. So, we don't think about the helpful aspect. So the idea of something being recessive is not necessarily negative, nor is the idea of what we call disease. We have to be very careful as evolutionary detectives. So when we look at malaria and sickle cell anemia, we're going to look at the upshot of malaria, well, the disease that causes sickle cell anemia in the community. I mean, what is its actual role in maybe protecting humans under the right context, so that we can think more critically about how evolution actually works, right? And how humans' conceptions of being good or bad may not exactly be right. All right, so that'll set you up to do laboratory 2. Okay, so after we finish laboratory 2, one of the things we have to think about is that there is a quiz coming up. And that quiz is going to be on September 24th. So as the date that I'm posting this is 2 weeks sort of in the future. 2 weeks in the future. Now, a lot of students will want to skip the next section, which doesn't have an actual lab attached to it. It's something I want you to actually watch. It's called Advanced Topics in Genetics. Do not. Because while there isn't, an actual physical lab I want you to do, there will be questions from the videos and the lecture, which I have. I have a PowerPoint lecture called Genetic Regulation that will feature prominently on the laboratory quiz that's actually coming up. All right, so don't forget that once you complete laboratory 2, go right down to that, the Advanced Topics in Genetics. All right, and again, last I want to say is that many students of you have begun to call me. I think you're getting more, accustomed to just pick up the phone and give me a call. And we've had some really good conversations with students that I have talked, and I think that it's really helped them. So whenever you come across any issues or any problems, even if you're doing a lab, the instructions don't make sense or the materials aren't there, you can't find them or whatever, call me, let me know. And quite seriously, I want to tell you that I do this for a living because I love working with students. And as much as this might be the worst times of lives for you guys because of COVID and all the social isolation, it's twice as bad for me. My whole life is about interacting with people, especially young people, to get you guys into a really good future, right? So don't discount my earnesty in wanting to work with you and help you on any issues in which you have. All right, so get started in laboratory 2. Again, any problems, let me know.